This video is made on behalf of Brian Corrie, Technical Director for the AIRR-seq project. Microsoft and Adaptive Biotechnologies are exploring how the immune system responds to COVID-19. As part of this project, they are gathering data from the adaptive immune receptor repertoire, also known as AIRR-seq data, from COVID-19 patients. The AIRR-seq data from this study will be stored in Adaptive's publicly available ImmunoSeq repository. In order to make this data more broadly available, there is a need to convert the data from the adaptive metadata format to standard AIRR-seq data format as created by the AIR-Community. The goal of this project is to develop a tool or a set of tools to query the adaptive ImmunoSeq repository using their web APIs, download data, and convert it to the standard AIR-seq data formats for repertoire. Once we have a converter for the adaptive metadata, it will be possible to load the COVID-19 data produced by the Microsoft Adaptive project and load that data into an AIR-compliant repository, such as the AIRR-seq turnkey repository. The AIRR-seq project will be operating a COVID-19 AIR-seq repository and will be working with Adaptive and Microsoft to curate this and other publicly available AIR-seq data once it is made available. This effort is in collaboration with and in response to the AIR-Community's call for sharing COVID-19 AIR-seq data. Once curated, the data will be part of the AIR Data Commons, making it accessible to the international research community in the fight against COVID-19. Not only that, but because the data is part of the AIR Data Commons, it will be possible for researchers to compare this data with other AIR-seq datasets from both healthy subjects as well as data from subjects with other diseases through searching and federating the data using the AIRR-seq scientific gateway. Sharing these data will be critical for developing diagnostics as well as therapeutics against cancer, infectious, and autoimmune diseases. Adaptive Biotech's approach to supporting their customers in tracking metadata about projects and studies that are in the ImmunoSeq repository is to be as flexible as possible. Adaptive uses both controlled metadata for some important fields and an extensible key-value tagging mechanism for other metadata fields. This makes it flexible for the researcher to annotate their study with metadata that meets their needs. Unfortunately, this approach makes it challenging for the data to be fair. In particular, a flexible and extensible tagging mechanism makes it for the data to be interoperable and reusable, as there is no guarantee that any two studies will use the same field name, definition, type for any given field. To solve this problem, the AIR-Community has established a set of standards for the curation and sharing of AIR-seq data. This includes the Maya AIR standard, which is recommended as a minimal standard for the curation of study, sample processing, cell processing, and nucleic acid processing metadata for studies that involve AIR-seq data. The challenge, if you choose to accept it, is to convert adaptive sample metadata file format into files that adhere to the AIR repertoire file format specification. Note that this is a non-trivial challenge, as there is no well-defined transformation for performing this conversion. Come join the AIR-seq adaptive team and make a difference in the fight against COVID-19.